So biotechnology, go figure, is using biology to produce a better product, to produce a better world. We are capable of changing our environment. We're one of the few organisms out there, us and beavers. We change the environment to suit us. And we've gotten to the point where we can not only change the environment, but we can change other organisms. So it's the application of technologies that involve the use of living organisms or products, or products from other organisms. That is, we use organisms to make other organisms. Milk, cheese, yogurt, beer, just about everything out there that we ingest is going to be a product of biotechnology. Now, we've gotten more advanced lately, but it's still the same process. We started off with domestication. Domestication is sort of the penultimate form of biotech. You take some living organism out there that had one role, and you change its role. Well, okay, so what was the whole point of, what did horses used to do before we got involved with them? They ran wild. What does that mean? They were rabid? They lived as they pleased. They ate all sorts of grasses, things like that. We got horses, and what did we do with horses? We ride them. We made them work. Now we don't have to do that work because the horse is doing it. Dogs. We took, dogs are like the ultimate story of human ingenuity. We took our predators and made them our friends. Not only our friends, but they do work. You guys, some of them do. Okay, your pets probably don't, but let's say like our Labrador Retriever. Anybody have one of those? No? Golden? Okay. You know what? Yeah. Those things will grab anything with their mouth and just carry it around, like for fun. We've engineered that into them. They didn't used to do that. They didn't bring them back. Cows. Can you imagine a wild cow?
I know there's no more wild cows anymore.
Right. But they came from wild stock. Right, the same thing, but we tamed these. Chickens. Again, imagine a wild chicken. Velociraptor, wild chicken. This is this is the baseline corn. This is what corn used to be. And then we got it, and we made it into what we know now. It used to be very, very small kernels, not very sugary, definitely not white, very short grass. No, it was terrible until we got a hold of it and worked on it for 1,000 years. And now it's all over the place. And it's only all over the place because we decided it was going to be. We've domesticated it. Just about every organism out there that you deal with on a regular basis has been domesticated. No. Well,
domesticating foxes.
Ah, well. So we've engaged in biotechnology for years and years and years and years. Now we're in this modern world of biotech where we can specifically genetically engineer microorganisms. We can make it very, very specific what we want to change. What used to take thousands of years of random mutation can now happen in a year or 2 years. It's still the same idea. Whether we're taking corn and it's getting mutated by ultraviolet radiation from the sun, or we're taking corn and mutating it through ultraviolet radiation in the lab, same process. Heck, we could even go faster. Instead of using ultraviolet radiation and having random mutation, we can actually make it site-specific. We can look at it and say, we want to hit that gene right there and see what happens if we mutate it. Same process, it's just happening a lot faster. A genetically modified organism, otherwise called a GMO, is any organism that's received genetic material through recombinant DNA technology. There you go. So anything that's gotten foreign DNA placed in it, and that's most things now. Chicken nuggets, whatever. I'm thinking, like, if you think about plants. Yeah, tomatoes. We've engineered genes into tomatoes to make them more resistant to cold. We've engineered, and to a lot of plants, we made them more resistant to cold so that we can continue growing them through the winter.
We figured out how to make tomatoes grow like not like a scattered vine, but go straight up.
Right. We've made things that used to be bushes into trees. We've made things into. We've made all of these leaps forward. Transgenic organisms, plants, and animals are going to receive genetic material from a different species. So there's a difference between genetically modified and transgenic. Genetically modified is the DNA has been altered. Transgenic is you've taken DNA from one organism and put it in another. If you were to take the green fluorescent protein from a jellyfish and inject it into a mouse egg, eventually you end up with a specific glowing mouse. Pardon? It does not attack it. It recognizes them as your own.
If you introduce it properly.
Right. It has to be introduced into the genome right early on.
And there's potential for failure. That's the whole.
There's a lot of potential. There is, for every success you have, you're looking at 1,000 failures. Now, this is obviously big business. Recombinant gene technology and transgenics really came in big in the late 1970s, early 1980s. That's when scientists realized they could do it. They can change genes. And in the 1980s, the court said you can now patent transgenic animals. So now you don't just have to let it, once you've created it, it's yours. That set of gene is your property, even if something else has the genes. You could sue somebody if they have a glow-in-the-dark mouse that used either your technology to build that mouse or they used the gene that you patented. And then you would get any product that they would have any money they would have been able to bring in. It is. That's exactly. But they've done that.
So how much money per year are you making on royalties from your?
Nothing, because I use somebody else's technology. I know. Now we can use microorganisms, genetically modified microorganisms, for lots and lots and lots of different things. We make medicine, fermentation, biological control procedures, and bioremediation. As far as medicine goes, we used to extract insulin from pigs. We don't do that anymore. We used to take the pigs, remove some of the blood from the pig, spin it down in a centrifuge, extract the insulin, and then use that to treat people who have diabetes. They stopped doing that about 15 to 20 years ago, and they switched over to having E. coli make it. Because instead of using pig genes, now we're using human genes. That's right, but we've engineered human genes into the E. coli. So it's transgenic. You're producing a ton of insulin now. Right, well, now it's changed slightly. It's very, very efficient. You're producing a ton of it. You're producing human insulin, which means there's going to be less allergic reactions. More people can partake, and it's a lot cheaper. As far as food goes, any microorganism that we use for food production, whether yeast in making beer, any bacteria used for fermentation, any when you're canning things, these organisms are, for the most part, genetically modified. If you find a beer, you're going to find that most of them have to be labeled as genetically modified because of the yeast that they use to produce it.
I've never seen an organic beer.
They've made them, and they're terrible. Biological controls. We can use microorganisms to control other microorganisms.
What if these latched onto it? Those aren't its offspring.
No, those are, so a parasitic wasp will have stung the caterpillar and injected its eggs. And then the wasp's larvae eat their way out of the caterpillar and then spin their own cocoons on it. They should stick to figs. But you can control these caterpillars, these hornworm caterpillars, through by introducing these wasps, these other organisms. That's biotech. Using bacteria to fix nitrogen. We spray bacteria on fields now because bacteria are nitrogen fixers. Plants can't do it. So if you have a field that needs a lot of nitrogen, like say a cornfield, instead of going and putting fertilizer out, you spray it down with bacteria. And those bacteria take nitrogen out of the atmosphere and produce what plants need. Exactly. Again, another example of, right, it's another example of biotech. In bioremediation, you take microorganisms and you could spray them down with the areas that we've screwed up. Right. We have not just mushrooms, but they've taken entire oil slicks. We have bacteria that devour oil. So we spray down an oil slick, and that oil gets degraded. There are beaches that have been utterly destroyed by oil spills. Black beaches. You've seen images of like otters and ducks and things coming up, and they're all covered in muck. We can spray these beaches now with a bacteria that will break down that oil into its constituent carbon components. So it goes away. Bioremediation. We've designed all of these organisms. You guys have probably heard of stem cell research, right? Stem cell research has a lot of potential to it. What a stem cell is a cell that has the potential to divide and divide and divide into any type of different cell. Totepotent ones arise right after fertilization. So back to birds and the bees, you have an egg and a sperm, and they come together and they fuse to form a zygote. After that zygote forms, it will start to cleave or split. So you go from 1 cell to 2 cells, 2 to 4, 4 to 8, and so on. And those cells can turn into any cell because they are sort of blank programmed right now. Eventually, they'll differentiate. But before that, they're known as totepotent stem cells. They have the potential to be anything, any type of cell in your body, including brain cells that you don't get to replace. So if you were to, the idea is if you were to take a stem cell that was compatible to your body and inject it near another cell, it will copy that other cell. It will make itself into a type of cell like that.
Is the DNA just like some sort of like a communication that goes on between them?
There is. You have to pass information between the cells. So you're going back and forth between the cells. So if one cell is going to be one thing, the cell next to it is going to, or the totepotent stem cell would adopt that shape. Would you be the same person? It depends. So, right, but, well, the issue is these cells are going to be slightly different than your cells because they're going to not be, unless you've got totepotent stem cells left in your body, which you probably don't, they're going to be somebody else's cells. Then that's different. And that's where you get ethical implications, when you start cloning, where people say, I'm going to create an organism exactly like me with my DNA that I will then harvest for its stem cells.
See, there's ethical ramifications either way.
There are.
Depending on what your perspective is. My dad needs some stem cell treatment for like an Lyme disease, but he won't do it because he doesn't believe in it.
There you go. So those are totepotent stem cells. Every single cell in a plant is totepotent. Every single plant cell can become any other plant cell. Humans and other animals are different. Typically, our totepotent cells disappear early on in development. They become fixed. That's where you get this idea of the embryonic stem cell. These lead to pluripotent cells, cells that can become almost anything. And this is probably what you hear about most in the news, where people are talking about embryonic stem cells. These cells can become nearly every type of cell in an organism. But the problem is, when you're harvesting them, you do destroy the embryo. So again, there could be ethical issues. That being said, we destroy a lot. I know it sounds really bad to say it, but we destroy a lot of embryos all the time, especially with in vitro fertilization. Well, it's not just that, but say a couple goes in to they want to have a child, so they go in for in vitro fertilization. They're not going to produce just one embryo and say, we're trying this. They're going to produce 12 or 15, because not all of them take. Well, once they have that, once you have a take, what do you do with the rest of the embryos? You freeze them, but then you have a maintenance cost year after year after year.
You can't just like give those to somebody else because it's not their egg.
You can't, right.
So
What
basically throw them away.
what ends up happening is they become medical waste for the most part.
And if you have permission, you can turn them into embryonic stem cells.
Correct. Now, less ethically questionable would be multipotent stem cells. You have lots of cells that are termed multipotent. For instance, you have stem cells that produce blood, hangs out in your bone marrow. If you lose it, you don't make blood cells anymore. So what do you have to get? You have to do a bone marrow transplant. Why do you need a bone marrow transplant? So you can keep making blood. Right. You've got stem cells that produce skin. They're constantly producing new skin. So we do have some multipotent cells, and in some cases we transfer them back and forth to each other if we're compatible. And then you've got unipotent stem cells. These are cells that are found in adults, and they can only become one type of cell. In your testes, well, not all of you, but in some of you, you have testes. These are going to produce sperm through your entire life. You've got a stem cell that keeps making more and more and more of this one type of cell. So these can be used for treating a lot of different diseases. Anything that causes deterioration, like Parkinson's disease, spinal trauma, joint damage, bone breaks, liver failure. These are sort of a medical almost cure-all. You can literally make new teeth. Yeah.
Do you like have to implant them or do they just grow?
No, you have to. Well, you have to inject them near a cell that is similar to the one you want to copy. So that's how lizards regrow their tails. Regeneration.
So in order to make a new tooth, you have to inject some stem cells into the old tooth.
Right. But then you could just implant that other one.
It would grow out of the tooth or
No, you have to plop it over.
take it and then insert it into the
Right.
nerve cavity.
This mini lecture really defined what biotechnology is, and it involves everything from anytime you use a living organism, ranging from cattle in the field, anytime we use ride a horse, domestication, any artificial selection we do in commercial crops, like choosing taller corn. All of our modern genetic engineering comes from those original processes of artificial selection, choosing one crop over another. What our modern genetic processes do is just speeds up how often these crops mutate, so that we can choose new traits that we're interested in. We can use genetically modified organisms for all sorts of things, including medication, food, fermentation. You can increase the rate of alcohol production, biological controls, and even bioremediation. In animal biotechnology, and even in plant biotechnology, we'd have stem cells. And these could be totipotent or pluripotent. They have the potential to be used to treat many, many different conditions. We may be able to take one cell and convert it into a different cell. So by studying stem cells, we might be able to understand how we can replace broken organs in our own bodies or dying tissues. These content review questions are here to focus your studies, to focus the information. In the next mini lecture, we're going to talk about techniques and how we're able to do biotech. And I'm not going to go into a huge amount of depth with them. I'm just going to give you an overview of the different techniques so that when something pops up on the news, you'll understand what technique was used and why they're using it.